From the Oregon State University Extension Service, this is Pollination, a podcast that tells the stories of researchers, land managers, and concerned citizens making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melathopoulos, Assistant Professor in Pollinator Health in the Department of Horticulture.
Astute listeners of this podcast will know that I have a deep and embedded passion for the breeding of honeybees. I find the whole topic fascinating. It's like unlike breeding in any other system. For one thing, most of the traits are expressed at the colony level, not at the individual level, making measuring those traits very difficult. In addition, it's hard to trace pedigrees. As many of you know, if you remember the episode with Julia Mahood, the mating takes place in the air. It's hard to control the mating. There's multiple fathers. It's really complicated. And that's where using molecular tools for identifying genotypes of importance are really important. I find the whole area of molecular biology, in contrast, very confusing, frustrating, and difficult to understand. And that's why I am so excited to introduce our guest, Robert Liu. Now, Robert is currently going into the second year of his Master's of Science in Biology at the University of Alberta. And I caught up with him in the Entomological Society of America meeting in Vancouver, British Columbia, where he won an award for a student presentation. It was very impressive. I was totally amazed at how he was able to break down some very complicated concepts into something that someone like myself could understand. He's in Olaf Ruppel's lab at the University of Alberta, and this week he's going to tell us about his research into breeding using these new molecular biology tools. That's how complicated it is, folks. Molecular biology tools to be able to select for virus resistance in honeybees, maybe one of the most complicated phenotypes that are out there. So this week in Pollination, we are going to dig deep down into the wonderful world of bee breeding with molecular tools and marker-assisted selection this week on Pollination. Okay, Robert, well, welcome to Pollination.
Great to be here. Thank you for having me.
Now, we're at Entomological Society of America. It is foggy. We're overlooking Burrard Inlet here.
Supposedly.
Supposedly. You can't see anything. But we're at the tail end of the Entomological Society of America meeting. You were in the student competition.
That is correct.
And it was really impressive. You gave a great talk, and it won an award here in the student competition, so congratulations.
Thank you. I actually did not know I won the award because, I was thinking, like you said, today is the last day of the conference. Well, of course the award ceremony will happen then, but it actually happened yesterday. And so I had no idea any of this was taking place until a buddy of mine who had attended, because he's more responsible than me and actually looked at the program, he texted me and was like, congratulations on your award. And I was like, what award? And it just sort of snowballed into me trying to figure out how exactly stuff went down.
Well, it was an impressive talk, and I think you did a really good job. For those of you who've never given, a scientific talk at a conference, you only get like 10 minutes to explain something intricate, and you had wonderful animation and elements to describe what we want to talk about today, breeding for virus resistance in bees. And maybe just to start with, to get our listeners on some footing, it strikes me that, there's some different, there's livestock breeding. We consider bees as a form of livestock. There's livestock breeding that's far advanced. I always remarked on this. You can pick up a catalog, and you know the expected breeding values of a straw of semen that you buy, and you know that the cows that are bred to that are going to have a few more pounds of this, and they're going to be that color, and you have these kind of detailed information, which was done over decades of data collection and pedigree data sets to be able to figure out what would happen if you cross this with that. And I know honeybees don't have that luxury because it's it's very difficult to control the crosses. So, but this whole area that you're talking about of genetic tools seems to allow us to leap decades over, and catch up a little bit. Can you just give us a brief overview of how you can use genetic tools to be able to infer the relationship of a trait to some underlying genetic thing that you can, select on?
Right, certainly. So I'm glad you brought up other livestock. Now, I should clarify. Technically, bees are livestock because we treat them as such. These are animals that were ferrying around to different crops, such as blueberries, and we release the bees, and they pollinate for us and produce our fruit. And in turn, they also produce honey, which is, as you know, a very sweet award for the beekeepers to go through the difficult process of managing bees and getting stung by them.
Yeah.
Which does happen, even with the protective clothing.
A lot.
A lot. More than I would like to admit. But yeah, it happens. But anyway, so the interesting thing about bees, maybe I shouldn't say interesting, but the reason why livestock breeding for bees in particular is so important is you have these large livestock animals like your mammals, such as your pigs and your cows and your birds, like your chickens.
Yeah.
Advanced vertebrates, as the taxonomists will sometimes refer to them. These are animals with very highly evolved immune systems that are able to adapt to infections by certain viruses. Mm-hmm. And in doing so, they're able to fight off these viruses, and we can give them medicine that helps them in this process. But bees don't really have that luxury.
Or you can vaccinate them all.
Exactly.
Yeah.
Exactly. And bees, unfortunately, don't really have this luxury. They have a very minimal adaptive immune response, as far as we can tell. Which is honestly already better than most insects that don't have an adaptive immune system. But primarily they rely on these cells of the innate immune system that just engulf anything that they find that they identify to be a foreign pathogen, for example. But a lot of these viruses are very good at circumventing these. Yeah, so a lot of these viruses are very good at circumventing these innate immune responses and make it very difficult for the bees to fight off. So a lot of the time these viruses are very severe. They'll just quickly kill the bees. And this is also compounded by the fact that in recent years there was a mite called Varroa destructor, which jumped in from the eastern honeybee, Apis cerana, into our western honeybee, Apis mellifera. And that brought with it a whole slurry of viruses which are now emergent threats that are rapidly propagating through colonies of the world.
Okay, so this is almost the imperative for breeding. So not only are bee breeding is kind of behind, but there's also these challenges that other livestock systems can deal with much more effectively because there's an immune system in them, whereas honeybees have to rely on. I mean, I guess the frustration that beekeepers have is they have these mites and these viruses floating around. There's almost seemingly nothing they can do about it.
Exactly, yeah. And we sort of have to rely on the bees to be able to kill off the viruses themselves, because we can't just give them some sort of medication like we can with cattle. We can't just dose them with antibiotics, for example. Not that that doesn't have problems in its own right, but it's certainly a more immediate solution than having all your bees die off and then trying to find a stock that doesn't just die off. So you're right, it is much more imperative that we breed resistance, or rather survivorship. I should choose my words carefully, and I'll elaborate.
Okay, so this problem is, we'll get into survivorship in just one second, but this problem is kind of the solution has appeared in terms of molecular tools that analyze the DNA and associate it with traits. Tell us how that process works.
Right, yeah. So, in theory, we know that if a trait such as survivorship when infected by a virus, if that trait is genetic in basis, then throughout a population we would see that at certain genes, variation in the individual's genotype of the bees will correlate with variation of the phenotype. So for example, you may have this one bee here with a particular genotype that correlates to high survivorship, or the ability to survive viruses better than, say, another bee with a different genotype that is much worse at surviving infection by some virus.
So it dies quicker.
Exactly. Or you have a population with different genetic compositions that a lot less of them die when a virus rampages through them than another population. It varies depending on how we look at the trait and how we define it.
The word trait and phenotype, they're synonyms.
Not quite. They're very closely related, but whereas a trait is a very general term of what characteristic we're looking at, the phenotype is how that trait expressed in an individual. So as a perhaps easy example, we can talk about something that most people should know about, Gregor Mendel and his pea plants.
Oh yeah, right. It's very famous.
Exactly.
Late 19th century monk who did these crosses and understood inheritance through looking at patterns of variation in these peas.
Exactly. So one thing that Mendel noted was that the color of the peas could be dependent on the parents that you used to breed that pea plant. And so you would have, for example, green and yellow peas.
Yeah.
Now, the trait that Mendel was looking at was pea color.
Pea color. Oh, but you had yellow ones and you had green ones. You had different. Those were the phenotypes.
Exactly. The phenotypes are the different ways that they express an individual. And likewise, genotype is different from genes in a similar sense. The gene is a functional region of the genome, this physical bit of DNA that does something. For example, in most cases, encoding for a protein, or I shouldn't say most cases, but in a lot of cases, it produces a protein. Now, this gene, just like the trait, can vary across a population, and how that gene varies is known as, the variations of the gene is known as alleles. And most organisms, well, not most again, but many organisms that we're interested in, such as bees, have 2 copies of the genome, each with one allele of a particular gene. And so the genotype refers to the particular alleles, the particular variants of this gene that are in an individual. So when I'm talking about genotype and phenotype, it's specifically referring to how the genetic and trait components of what we're testing for present within a individual organism.
Okay. So we got that out of the way. So how do you? I imagine you can't just go with a microscope and look at this variation in these in the genetic sequences of these individuals that are exhibiting different survivorship. So how do you find them?
Yeah, I mean, in theory we could do that, but well, I would say it would take you forever to, but reality is that you'd run out of money before you were able to screen every single gene in the genome to try to find one that correlates to whatever trait you're looking for. So we can do something called quantitative trait loci analysis, or quantitative trait locus analysis. Locus is just the singular word for loci. And what a locus is
it an allele?
Not quite.
Okay, all right.
To clarify, locus is not the grasshopper that swarms and eats all the different plants. It doesn't have the T at the end.
Like a location.
A locus is a particular location. It's somewhat arbitrarily defined based on our purposes, but it's essentially just a region of a gene. So an allele or a gene could certainly be a locus, but generally we take larger chunks of the genome as loci.
Okay, we just break this down though. Why is it quantitative?
Well,
and what's the trait have to do? There's 3 words in this thing that you told us about.
So a quantitative trait is a trait that expresses quantitatively throughout a population.
Survivorship. It lived 14 days. It lived 24 days. It's a continuous quantitative measurable trait.
Exactly.
Gotcha. Okay.
And so the loci that correspond to that quantitative trait are these regions of the genome that will vary and that variation will correlate to the variation of the trait. Now, how do we know whether a locus varies or not? Well, we can use these things called genetic markers, which are essentially regions of non-coding DNA. So DNA that essentially is junk DNA, doesn't do anything, but
There's junk DNA?
Yeah, in fact, most of our genome is junk DNA.
Really?
I believe something like 50% or more is just one type of junk DNA. Called a transposon, that is essentially a gene that replicates itself and then inserts itself into another region of the genome. It's very, very interesting stuff.
But if it's junk, then selection it's stable then. Is that the idea?
Well, the idea is that if it's junk, it actually is unstable because it doesn't really matter whether or not it mutates, right? It's not like, for example, the gene that controls oxidative phosphorylation, which is the process that happens in the mitochondria, the powerhouse of the cell, everyone. If that gets mutated, the organism is finished because it can't produce energy.
Oh, so there's no consequence. So if a mutation happens, it's just drifting along in history.
Exactly, and what's great about that is that there's a lot of variation within a population at these regions, which make it very easy to tell individuals apart by their markers. But it's also very easy to tell different markers within the genome apart. And so we used a type of marker called a single nucleotide polymorphism, an SNP, which is just one base pair of DNA gets mutated. And that's a very common variation because it's such a very simple mutation that can happen.
Okay.
So when we track the variation of the quantitative trait loci, and we see that one particular locus, or I should say when we track the variation of loci, and one particular locus correlates to a trait that we're looking at, we call that locus a QTL, a quantitative trait locus, because it's statistically related to the quantitative trait that we're measuring. And the relevance of that locus now is that we can infer that somewhere on that region of DNA, there's probably a gene that encodes for the trait we're looking for. Now, to be fair, a single locus can still contain hundreds of genes, but that's a much smaller amount of genes that we'd have to screen through than, say, the millions in a full genome.
Is it significant that it's so small, the SNP, the single nucleotide polymorphism, that it's so small, it's like at the finest scale? Is that afford some kind of functionality when you're looking for traits?
So when you say functionality, do you mean?
When you're trying to find the needle in the haystack, is it useful to have, to help me wrap my head around the concept?
Yeah.
To have something that's so fine, very fine, as opposed to like a very kind of bigger region.
Well, so modern genetic techniques are very good at picking up fine scale details like this, and especially something like SNPs, where, yes, you're right, they're very small and it's very hard to hone in on one, but because they're so small, they're also all over the place.
So it gives you a lot of resolution then.
Yeah, it's not quite the needle in the haystack sort of situation. It's more like you have a whole bunch of needles in a haystack.
And you know where they are.
You don't necessarily know where they are, but you can infer that if you put your hand in, you're going to get poked a fair amount, to say.
All right.
And so, obviously, we're not looking at every single SNP that exists in the genome. We can't pick them up to that level, but we can still quite easily pick up tens of thousands. And in fact, our data set, which I'll talk about in a second, we had exactly that, tens of thousands of SNPs. But
well, before you go on, so
yeah.
let me just see if I got the concept. You got these two bees, two populations of bees, or two colonies.
Yeah.
You've got similar genetic background, and you give them some virus, and you notice that these ones die 3 days earlier than these other ones, and so you know that the virus, these ones have a shorter survivorship or a longer survivorship, and then you go in and you extract out their DNA, and you're looking for these small. A lot of the DNA there will be a lot of DNA that has these small little variations. So this one is, you look at the two bees, and this one's slightly different than this one, and so which allows you to sort of like have all these needles in the haystack, and then what? Did I get the concept right there?
yeah,
Okay.
exactly. Yeah, and then we take a look at each of these SNPs. We call them SNPs, which is probably easier to say than SNP anyway.
SNP, yeah, okay.
SNP. And we track their variation across all the bees that we sampled, and then we statistically compare them to the variation of the trait that we're looking at.
Okay.
Now, one thing to note is that despite the method we use being called quantitative trait locus analysis, QTL analysis, it actually can be used on binary traits as well.
Okay.
So if we're looking at something, say, just able to survive infection or not able to survive infection, that's a very binary trait. It's not quantitative the way we would traditionally think it would be, but that's actually not a problem. We can still, the concept of, okay, well, the genotype correlating with the trait, that still exists. That correlation will still exist no matter whether we consider the trait to be binary or not. We just keep the terminology the same because that's how it was originally developed. Now, the advantage of QTL analysis is that you actually don't need a very large population. You can just do a self-contained cross where you take bees of different parentage, you breed them together, and you create a hybrid bee that you then cross with one of the parents, and then you have a whole bunch of variation in the lineage of your resulting bees, and you can use that variation to directly compare to the phenotype that these bees are expressing.
So let me get this straight. So you've got variation in the phenotype,
Yeah.
but then you have variation in the which SNPs are associated with that trait, with that phenotype, and that allows you to sort of infer a relationship.
Right. Yeah. So the variation is going to be present in all the different SNPs, but we're mostly interested in SNPs that vary in correlation with the trait because that is the association that we're looking for. So in our experiment, I think it's a more concrete example. For example, the experiment that we did. Now, this breeding part was actually not done by me. It was done by our collaborators at the USDA. But what they did was they had a whole bunch of colonies in the bee yard, and they took a bunch of worker bees from each one. And after that, they dosed each bee with a certain amount of virus. In our study, it was a virus called Israeli acute paralysis virus, which is a virus that was a great tool for us because it's a very severe virus which expresses the survivorship very quickly. Bees will really quickly die within the span of days. If
with some viruses it can be latent or it doesn't express under certain conditions, but this one's reliable.
Exactly. It's generally very quick if it takes hold of a bee, and if it doesn't, then after a few days you'll know whether your bee survived or not. And it's also found in lab grade samples, so we had a bunch of pure virus that we could work with. Now, what Mike Simone Finstrom at the USDA did was he dosed each bee with IAPV, Israeli acute paralysis virus, and he waited for 3 days to see how many of the bees from each hive dies. From the hive with the lowest amount of death, he took a queen, which he labeled as the resistant queen. And from the hive with the highest amount of death, he took a drone, which is a male bee. And then he extracted the semen from the drone and artificially inseminated the queen with it. Interestingly enough, we can't seem to figure out how to naturally mate queens and drones, so artificial insemination is currently the method.
But that must give you a lot of control because you can get, in the wild, I imagine multiple males mate with a female, but here you can get one single male inseminated, so you've got a perfect cross between that one line where they had low survivorship and high, and make a progeny that are some kind of mixture of the two.
Oh, exactly, yeah. And the upside of that is certainly, the control. But the downside is if you're trying to cross a whole bunch of bees, it's a lot more work.
Technically hard, yeah. Okay.
But getting back to the study, so this would have resulted in a hybrid queen, in theory, which would have half of her DNA from the susceptible colony and another half of her DNA from the resistant colony. So this bee was then bred back with a susceptible male, because we think that the resistance genes are going to be dominant over the susceptible genes. And so what we did was this is called a backcross, because her father was a susceptible phenotype, and now her mate is also a susceptible phenotype. And so what the result is a bunch of worker bees who their fathers and the chromosomes they inherit from their fathers are all susceptible. But from their mother, there's some meiosis going on, some recombination going on when she's making her eggs, and this scrambles the genetics of the resistant and susceptible lineages.
Oh, I see.
Which then gives us a very heterogeneous makeup in our worker bees in terms of their maternal genetics.
So let me get this straight. So the goal of doing all of this crossing extremes and then back crossing is to really create as much, to kind of maximize the variation available in that pool.
Exactly. We want to create a very diverse set of genetics, which in turn, hypothetically, if our hypothesis is correct, will create a very diverse set of phenotypes.
Okay.
So with these bees, another collaborator of ours, Shilpi Bhatia, who was at the University of North Carolina, Greensboro at the time, she took a bunch of these worker bees and she again dosed them with IAPV. And after that, she waited 3 days to see which ones would die and which ones would survive. The bees that died were considered susceptible, and they were collected. And the bees that survived were dosed again with IAPV just to increase the amount a little bit. And then we waited another 2 days. And those that died were considered intermediate, while those that survived were considered to be high survivorship. We called it resistant at the time, but resistance has a very strict meaning, and I'll get into that a little bit later. But they were high survivorship. They were survivors. Now, we took the survivors and the susceptibles, and we collected them. We actually, though, discarded the intermediates. And the reason why is because we want to compare the two extremes and say, okay, what's going on with these really hardy bees that these very susceptible bees don't have in their genetics. And so we sequenced them throughout their entire genome just to try to find as many SNP markers as we could. But we also collected a tissue sample from each bee. And that tissue sample was used to check for the presence of any other viruses. You know, we're measuring our survivorship based on death. But we're also assuming that the death was caused by the virus that we're dosing them with.
Right.
And the problem is, if there were any other viruses within that population of bees that were causing death, that would confound our experiment. That would screw up our data.
Okay. So you take this sample out and you can analyze it for viruses. You've got these two extremes based on the test. You've got the colony and you just take some subsample of workers out, you feed them some virus, and you confirm that these ones die very quickly and these ones die very late. So you've got these pools. Okay, you got two sets of samples. What then?
Well, then we sequence them at their SNP markers. We take the tissue sample to check for other viruses, and then we run our QTL analysis. Now, there are technically many different mathematical models you can use for your QTL analysis in order to find these quantitative trait loci. The simplest of them is something called a single marker analysis, where you take each SNP, each genetic marker, and you compare them directly. You compare the genotype at that SNP directly to the trait. And the idea is that each SNP represents its locus that is found on, which is bordered by other SNPs. Now, this can be done with a very simple statistical test that just gives you a p-value, but the problem with this test is that these SNPs are not as evenly distributed as we may want them to be, and they're not as tightly packed together as we may like them to be. Sometimes you have large gaps between your SNPs, and if you're only using the SNPs as markers, then anything between those two SNPs where there's a large gap, we can't infer anything about what genes might be going on there. And also, genotyping is very good. Like sequencing is very good, but it's not perfect, and we have a fair amount of our SNPs as missing data because there was just something wrong with the sequencing that went on during that time. And that's also lost data, right? Because we can't use a SNP if we don't know what the genetics at that SNP are.
Okay.
And so we can use a different model called interval mapping. And what interval mapping is you use the genetic markers to create a genetic map, which you can use many different software for. Most of the time, QTL analysis software has that function built in. Now, with this genetic map, it sort of simulates what's going on in these large gaps using what information is already available.
Oh, I see.
And in doing so, we can then have essentially a perfectly covered genome that we can then subdivide into
I see.
intervals.
So where the single nucleotide comparison is kind of just looking, it's agnostic to position and all of these things. This interval mapping kind of like thinks about how they are related on the genome, and that allows you to kind of say, well, I know Calgary, I know that Corvallis lays between Eugene and Portland, so that if the trait is somewhere, north of Corvallis but south of Portland, you'll know that because you can look at the...
Exactly. We're essentially inferring what we cannot directly observe using what evidence we have, which, to be fair, is a lot of science anyway, right? But yeah, this method is it allows us to get a very fine resolution of what's going on in the genome, but it's, as you can imagine, very computationally intensive for the computer to take in all of our markers and spit them out. And most of the time with QTL analysis, you don't have more than, say, a couple hundred markers.
Yeah.
We had 54,000.
Okay.
So I tried putting that data set into my computer, and it basically said, no, I'm not doing that. So what we did then was we came up with a bit of a solution, which was something called consensus marker creation. Now, what this essentially means is that we took groups of 20 markers from our raw data set of 54,000, and we used a majority rules algorithm to say, okay, most of these markers say that they're coming from the resistant lineage. Some of them say they're coming from the susceptible lineage, but majority rules. We're going to say that this marker is this genotype that relates back to the resistant grandmother. This allows us to really concatenate our data set into a data set of 2,700 markers, 2,700. Really big, but a lot more manageable than our. But without getting too into the specifics of measurement, we were able to then create this map of the full genome that represents a full genome that is made from these majority rules markers. And then we could trawl this full map for the presence of any quantitative trait loci, any loci that are statistically correlated to our trait. And once we did find a particular interval or a particular locus that did correlate to our trait, we could then go back to our raw data set, take that chromosome that we found that QTL on, and use the single marker analysis on that chromosome to find out exactly where this peak would be in the real genome. And that would allow us to do things like, say, we could go into a gene browser to figure out if there are any genes within that region that might be, say, an immune system gene or something else that would allow us to specifically breed for this survivorship against this virus.
Okay, let's take a quick break, and then we'll come back and want to hear what you found. Okay, we're back. We were just talking about risky science, which kind of leads us into your findings. So you've got these traits mapped using these very ingenious methods, and making a compromise between computationally complex but accurate, sort of, gives you more coverage and, using a rubric to try to adjudicate this. So you end up with, so tell us what you found.
Right, so just to
Did they map perfectly? Did you have a bunch of, I imagine, loci that were just so tightly linked to the trait that they just shone up like a marquee at a downtown Broadway
so
play?
Just to quickly recap, what we did was we sequenced bees at a bunch of different markers in their genome, and then we used those markers to create a simulated genetic map, which we then subdivided into individual sections. And each of these sections, we compared variation across our bee population in that section to a variation in survivorship when infected with IAPV.
Okay.
And the results were just not. we did not find a single QTL that was significantly correlated to virus survivorship of IAPV.
Does this, I guess at one point, like we were joking at the interval, like that, it's like, but does this mean that you can rule out that there's one strong trait that governs this? Is that one of the things you can infer? If it was hard to find using such a rigorous methodology, that it's unlikely that there's like a uber trait out there?
Now, when taken at face value, the answer would technically be yes, we could rule it out. However, before we conclusively say that, we have to go back and look at our experimental design. I think is important. It's always important to scrutinize how this experiment was created, because we have to keep in mind that an experiment is not necessarily reality. It's a model of reality. And so when an experiment tells us one thing, we can't say this is what the reality of it tells us. We instead have to say this is what our model of the reality tells us, and we proceed with how we apply that model to reality. So in our situation, our model had the underlying assumption that, okay, well, this colony had a lot more bees survive when dosed with IAPV than this other colony. That means this colony is much more susceptible than the susceptible colony. But if you actually look at the data that we had, the non-susceptible colony had 70% surviving, while the susceptible colony only had 50% surviving.
Resistant.
The susceptible colony had 50% survivors, while the quote-unquote resistant had
so
70.
it wasn't, the thing is that perhaps the experiment didn't really capture the full breadth of variability that exists in the bee population.
Exactly, and I think with studies like this, ideally you want to have a lineage which you've bred some more in order to really homogenize exactly what traits, what genotypes it has, which in turn mean that all your traits that are related to those genotypes should also be very homogeneous. And well, 30% dying to this very severe disease is actually reasonably good survivorship. It's not really that great.
You can't rule out that there is, because you may have not had those alleles for very high survivorship in the population to start with, so you wouldn't be able to detect them because they weren't there.
Exactly, or it's entirely possible that they were there, but they were quite distributed they were also distributed within the susceptible population, and so we wouldn't be able to disentangle the associations.
Right, because you hadn't really kind of concentrated it, and so those traits may be confounded because you haven't. They're still. With increasing breeding, they would have left the susceptible population.
Theoretically, yeah. Or we would have ended up with another resistant population.
Okay.
That would be, and that is also how a lot of the breeding has been done historically, is you don't actually know what the genes that you're selecting for are. You just see that, okay, well, this bull is very resistant to some disease that they get, and this cow is also very resistant. Let's breed them together, and let's take the healthiest of their young and let's breed them together again. And that's how a lot of the selective breeding occurs. And of course, we're trying to streamline it, so we're not doing that. But this is one of the problems, is we don't have these very nice homogenized populations to work with. These purebred lines.
Well, just we should probably wrap up here, but just to return, there was silver lining. So you did, fortunately, somehow you thought to take those samples to see what viruses were in the bees, and how did you use that to sort of...
Yeah, so I'm glad you brought that up. For those who've forgotten, remember that we took a tissue sample from each bee that we tested to screen for any other viruses that could be killing bees. And we didn't find any other viruses that were high enough levels to kill bees. But we did find that for some of these viruses in our sample, we had 8 viruses in total, and for some of these viruses, there was a lot of variation in the amount of virus that each bee was carrying. And this is despite the fact that they all came from the same hive. So we thought, let's do a QTL analysis on these viruses as well. And for 3 of them, specifically deformed wing virus B, Kashmir bee virus, and Lake Sinai virus, we found that there was actually quite a significant correlation between what genetics the bee had and how much of this particular virus that they were carrying. And this actually was a quantitative trait, so it truly was quantitative trait analysis in this case. Now, it doesn't necessarily mean we can infer anything from these results, because we didn't design our experiment specifically to determine the relationship between some genetic element and these viruses. It was just sort of a flash in the pan, sort of, oh, this is interesting, we should look at it. But to that end, I think if I were to just throw out what I think is going on, or what I don't think is going on, rather, what we noticed with these viruses is that the genes that were relating to the amount of virus that a bee had were on different chromosomes. And so, like I said, most of the immune system of a honeybee is just very general. It just sees something that it recognizes as a pathogen and destroys it. Because these genes were on different chromosomes for each virus, we would have to infer that these are probably not the one-size-fits-all immune genes that are reacting to these viruses. Rather, it's something specific to the virus. What that is, we don't quite know. We would have to carry experiments similar to the IAPV experiment using well-bred lines, and that may be possible sometime in the near future with better characterization of some of these viruses, but for now, our abilities to test such a thing are limited.
Well, that is fantastic. I love, one thing I really appreciate about your work is, just the kind of doggedness, and also, in the face of results that were, maybe not the, what one had hoped for, being able to kind of process through and think about the next set of questions as coming out of such a set of results.
Now, I was doing one thing that you may have noticed throughout this talk. I was stopping you when you said resistance to jump in and say that perhaps not resistance. And I was actually talking to one of my colleagues. She's a fantastic scientist in her own right. Her name is Nina Sokolov from the University of California, Berkeley. And sorry, did I say Sokolov? I meant to say Solikov. I'm sorry, Nina. Nina Solikov.
It's been a long meeting.
I'm honestly not good with names. Faces, yes. Names, no. But Nina Solikov. Nina Solikov.
Yeah.
So she is looking at virus transmission from honeybees into wild bees, which is actually a very big problem because it's the wild bees that are really suffering right now. So one thing that she wanted to bring up with me that we followed up on, we had a very good conversation over coffee yesterday, was that when we look at bees surviving a dose of virus, we can't infer whether or not this bee is, strictly speaking, resisting the virus, in that it is suppressing the viruses that are getting into them and preventing them from establishing, or if they're perhaps just more tolerant of higher virus loads within themselves.
So they don't stop the virus from multiplying, but they kind of like go about their business even though they've got lots of virus.
Exactly, and the viruses just aren't affecting them. Now, functionally, if you're a beekeeper, no difference, right? You have less bees dying, you can have more virus, you can have better output.
But I imagine, you may have bees that are packed full of viruses and transmitting them to something that's susceptible.
Exactly. If we have higher tolerance for this virus, then these bees are going out and they're essentially virus bombers that are dropping viral loads all over the environment, and that's just more danger for the native bees that also need our help. And so I just want to conclude this talk by saying that these results are very preliminary. They are exciting, however, and there are very many cool implications. However, I think we need to approach these results very carefully, because when we breed for livestock, I think it's too easy sometimes to get fixated on this system that we're working with to forget that all around us is this big interconnected ecosystem, and what we do with one thing could have huge impacts on everything else in the ecosystem. So science is cool, but it's also a very tricky thing to get right, I guess.
Thank you for helping us navigate through the trickiness of this whole field, which I think you've done a wonderful job explaining for myself and also for my listeners. So good luck with the rest of your research.
Well, thank you. Thank you for having me. I'm very happy to just be able to educate the public on what I think are very important issues.
They're very appreciative.
Thank you.
Thank you so much for listening. The show is produced by Quinn, Sin, and Neil, who's a student here at OSU in the New Media Communications program, and the show wouldn't even be possible without the support of the Oregon Legislature, the Foundation for Food and Agricultural Research, and Western SARE. Show notes with links mentioned on each episode are available on the website, which is at pollinationpodcast.oregonstate.edu. I also love hearing from you, and there's several ways to connect with me. The first one is you can visit the website and leave an episode-specific comment. You can suggest a future guest or topic, or ask a question that could be featured in a future episode. But you can do the same things on Twitter, Instagram, or Facebook by visiting the Oregon Bee Project. Thanks so much for listening, and see you next week.